So the Agrobacterium infection process that I was talking about last time, it initiates with a damage to the plant cell. And Agrobacterium should be actually in the vicinity of that damage if this infection has to take place. The damage to the plant cell results into breakage of the plant vacuole, which releases acetylsyngine, which is the phenolic compound, and then the release of vacuolar contents. They also decrease the pH of the environment to about 4.5 to 5. Okay, so this is a condition which is required by Agrobacterium for infecting uninfected plant cell. Okay, so what happens is that this decreased pH and acetylsyngine, it triggers the Vir-A gene, which is code for Vir-A protein. And Vir-A protein, actually, it is constitutively expressed and is present in the cell wall of the Agrobacterium. Upon this activation by acetylsyngine, this Vir-A protein gets phosphorylated, which in turn phosphorylates another protein which is constitutively expressed and is also available in the cytoplasm of Agrobacterium, that is Vir-G protein. The phosphorylation of Vir-G protein triggers the expression of the rest of the Vir genes, all the other Vir genes which are involved in tDNA synthesis, in movement of the tDNA from the bacterial cell to the plant nucleus. Okay, and then in the plant nucleus, this tDNA gets double-stranded, which is originally actually single-stranded when it is produced from the double-stranded TI plasmid. Okay, so after getting double-stranded, it gets integrated into the host genome through some recombinational mechanisms which are governed by the terminal edges of the left and right borders on this tDNA. Okay, upon integration, whatever is there in this tDNA fragment, whether if this is a natural TI plasmid, it will start expressing, this fragment specifically will start expressing the genes involved in synthesis of the opines and the genes involved in the cytokinin hormone production and auxin. And that these two hormones actually, they result into the formation of tumor on the plant cells, which is usually known as crown gall. Okay? And in recombinant DNA technology, we displace these genes for opines, cytokinins, and auxins with the genes of our interest. When I talk about genes, make sure that these are the gene cassettes. Okay, so a promoter, a gene, and a terminator represents one gene cassette. So you can put 1, 2, or 3 different cassettes on this tDNA using the synthetic TI plasmid vector. Okay, and those genes, they will make actually the cDNA, the tDNA, and the tDNA will migrate to the nucleus, incorporating your genes of interest, and the plant will start expressing those genes of your interest, whether they are for insect or pest tolerance, or they are for salt or drought tolerance, whatever the genes are of agricultural importance. Linked to this slide is another slide which tells you about the mechanism of this movement of the tDNA from the bacterial cell, which is here, okay, to the plant cell, which is here on the right-hand side, okay. So you can see here that if there is a wound in the plant cell, The plant signals like acetylsyringion and lower pH, they activate Vir A and Vir G. Vir A is there in the cell wall, it gets phosphorylated. Vir G is in the cytoplasm, it gets phosphorylated, and this in turn produces a lot of different virulence genes which are present on the fragment of DNA which we have in our TI plasmid. Okay, all these genes start expression. And then if you remember the VIR-D2 is the one which gives a single stranded cut in TI plasmid, generating a single stranded tDNA, okay, between the left and right borders. And this tDNA then moves into the plant cell through a set of proteins. These proteins are not made as a result of these virulent genes, but they are usually associated with most of the gram-positive, gram-negative, and RT. All these cells, they have this type 4 secretion system, which is based on the conjugal transfer of DNA or proteins in the surroundings of the cell or towards another cell which is attached to the bacteria. Okay, so in this case, there is an attachment between Agrobacterium and the plant cell, and these proteins, which are known as important proteins. Okay, so they take this tDNA along with the VIR-E2, which has already bound to the single-stranded tDNA, and it gets into the nuclear membranes, and then where it is made double-stranded. Remember that the single-stranded tDNA is made double-stranded before it gets incorporated into the plant chromosomes. Usually what happens is that the transformation by Agrobacterium results into the formation of just one tDNA per bacterial cell. And the bacterial cell can then attach to a plant cell, usually one bacteria, one plant cell. Okay, so there is a transfer of single tDNA from the bacterium into the host nucleus. So there are not multiple number of tDNAs entering into the plant nucleus. And this results into a single integration event. That because there is a single tDNA, single insertion, single event. Only one cassette or two cassettes or three cassettes present on the same tDNA get incorporated, but there are not multiple tDNAs which are incorporated in the nucleus. This is actually the most frequent case, and that's the reason most of the time that you infect the plant cells with the Agrobacterium at a very specific optical density. Okay? That is usually 0.4 to 0.5. At this density, the infection process is very efficient. Okay? The bacterial cells, they are fastly growing, and they are all their activities, they are so much functional that they can easily complete this process to the end. Okay, so this was the mechanism of integration of tDNA into the host genome. Okay, so single tDNA on any of the chromosome at any of the location. So all this integration is a random integration of the tDNA. Okay, that at what position the tDNA gets inserted into the chromosomal DNA, it cannot be controlled by the humans. Okay, so this is all natural process. Now I mentioned that there are some ATT genes coded by the virulent region of the genes on the TI plasmid, and these ATT genes, they are involved in the binding of this Agrobacterium on the plant cells. Okay, so here this is an electron micrograph that how the Agrobacterium attaches to the plant cell. Okay, you can see that it's a kind of polar attachment. Okay, most of the cells, they are attached to on one pole of the bacteria. This is gram-negative bacteria. Okay, so it produces some microcellulose fibers which help to attach this bacterium to the plant cell, from where the tDNA can then move into the plant cell. So it is a kind of attachment through microfibers of cellulose. Okay, this is a very clear picture of that how does the agrobacterium interact with the plant cells. Now, previously I mentioned that some of the bacterial strains, they are Nopaline type, some of them they are Octopin type. Okay. The Octopin types, please turn off your mic. Some of the bacteria which are of Octopin type, they have two tDNAs produced which migrate to the host cell, okay? And there are two integrations. And those tDNA formation is supported by an overdrive sequence. Now, the virulent genes, which are spanned over 40 kilobase fragment, some of those genes, okay, they are responsible for host specificity, okay? Some of the genes, they enable the bacterium to infect one kind of host, while the other genes, they enable the bacteria to infect some other kind of host, okay? So any TI plasmid can be a wide host range if it has got the genes, a number of different virulent genes which enable its capacity to infect wide type of dicot plants. Or it can be a limited host range if the number of these host range genes are limited to a few, okay? So the bacterial strain which is carrying this TI plasmid can be a wide host range infection process, or it can cause a limited host range. But there are some strains of the Agrobacterium which are called super virulent strains. And I mentioned it last time that if you take out the 40 kb fragment from TI plasmid and insert that fragment in the genome of Agrobacterium, the host is now called super virulent strain. The reason is one set of the virulence gene is present on the bacterial chromosomes, the other set of genes is present on the TI plasmid. So all those genes, they will be expressed at a very good efficiency, resulting into increase in the host range of the bacteria. And this is the reason that initially none of the Agrobacterium strain could infect monocot plants. But when they are made super virulent strain by introducing the Kamuri fragment, which is actually the 40 kb virulence genes present on a natural TI plasmid, okay? So those strains, they become super virulent and they can transform a greater variety of plants like monocots too, okay? So Agrobacterium transformation is now used both for the dicot plants and the monocot plants. Okay, the super binary system, which I will introduce you shortly, what is the binary system. It has also got extra set of Vir-G, Vir-C, and Vir-B genes on it, okay? You know that this is activated, which controls the expression of all the Vir genes. Vir-C binds to the overdrive sequence, and Vir-B is another protein which is involved in the transfer of tDNA to the plant cell. Okay? So if we take all of these genes together as extra copies in the TI plasmid and the host chromosomes, the system becomes highly efficient for transformation into the plants. Okay? So if we look into the transformation vectors, the basic transformation vector, which I mentioned last time, I asked you the question that what should be there on the TI plasmid? It must contain the three basic features of any plasmid vector, which is used for general purpose cloning vector. Okay? Those basic features are the origin of replication, an antibiotic selection gene, and a multiple cloning site. Those basic features. But a TI plasmid should contain additionally a left border and a right border. So antibiotic resistance gene, on one side, it is present outside the left and right border for the selection of the plasmid within Agrobacterium. Okay, but then we also need to do the tDNA integration selection. So the tDNA must contain another kind of antibiotic gene that we can use for selection of the calli which is infected by the Agrobacterium strain. So the selectable markers mostly associated with TI plasmid, they are hygromycin gene, gentamicin, bar gene, that is BASTA gene, that is insecticide resistance gene. Okay. The first thing you need to do when you join Zoom is to turn off your mic. This has been repeated so many times. EPSPS is another herbicide tolerance gene. Okay. So all these genes can be used as a single marker gene. You can select any one of them in the region which is between the left and right border. Okay, and this selection of the antibiotic resistance helps in identifying the plant calli which have got the tDNA inserted into it. Okay, the selection is based on the antibiotic or the insecticide resistance gene that you have already incorporated in the TI vector. So you don't have to do it. It depends what kind of TI vector you select. And look into the gene, antibiotic resistance gene, which is there on the tDNA region. Okay, and then you can use just the MCS site to clone your genes of interest. Introns. Sometimes what people do is that for expression of a gene in a eukaryotic system, they insert introns. They use the genes having introns, or they can incorporate an intron between the promoter and the gene. Okay, which actually, it's not really true, but it is reported that insertion of the intron within the genes or insertion of an intron after the promoter thus increases the gene expression. Okay, so sometimes people use introns. Some other stuff that can be used for efficient transformation of the tDNA is the scaffold attachment regions or matrix attachment regions. This is a bit complex, actually, The chromosomes, when they relax within the nucleus, they have some outer boundaries. The regions of the DNA which are most relaxed on a chromosome, they are attached mostly with the nuclear membrane. And those are the regions which are said to be highly expressed. Okay? So the chromosomal DNA is relaxed from the chromosomes, attaches itself to the nuclear membrane, and the expression of the genes in that vicinity of the DNA is highly dominant. The sequences which help the DNA attach to the nuclear membrane, they are called matrix attachment regions or scaffold attachment regions. Okay. Sometimes people attach such regions in the boundaries of their genes, the gene cassettes, so that these regions can relax the DNA over there and attach them to the nuclear membrane so that the relaxed part of the DNA can be easily transcribed to give the messenger RNA. Okay, so just keep in mind that these are the regions on the DNA which help in better efficiency of transcription. Okay. So sometimes people add such kind of things, okay, but it is not for sure that they really, some people report it works, some people report it does not matter, okay. Now I'm coming to the binary system. If you remember in a slide here, I mentioned super binary system, okay, for monocot transformation. What is super binary system is? Binary means two. What you do is that currently, either for the transformation of dicot plants or for the transformation of monocot plants, we need binary vectors. Binary vector means we need a TI plasmid and a helper plasmid. What an helper plasmid has got? All the features of the plasmid, origin of replication, antibiotic selection marker, but no multiple cloning site on it. Okay, the tDNA is deleted. It means there is no left and right border on it. What it has got? It has got the 40 kb virulence genes on it. Okay, because there is no virulence gene on the TI plasmid. Why? I said the original TI plasmid vector is 200 kilobase big, and the one we use in laboratory for transformation is about 10 kilobase. We have reduced the size so that it can accommodate our genes of interest. 10 kb, 20 kb of our gene of interest. If this is a bigger plasmid, then you can think that how difficult it will be to transform it and then to make the tDNA from a large region. Okay, so this plasmid, which is TI plasmid, is really, really small. It is 10 kb, having left border, right border. You can see there is a kanamycin resistant gene between the left and right border, and there is a multiple cloning site because the tDNA is produced between left border and right border. Okay. So this part of the plasmid, on the other hand, contains origin of replication, and it should have actually one antibiotic gene other than kanamycin on it as well. Because when this plasmid is transformed in Agrobacterium, we select the transformed Agrobacterium based on the antibiotic resistance gene on the TI plasmid. Okay. Now I told you that the helper plasmid carries the virulence genes. What it means? When we do Agrobacterium transformation with the TI plasmid, it's not a single plasmid that we are transforming. We are transforming two different plasmids. One is the TI plasmid, the other is helper plasmid. TI plasmid does not have virulent gene on it. So those genes are supplied by this helper plasmid. Okay, so the where G get phosphorylated and start transcription of these genes. And these proteins which are produced by helper plasmid, they help to produce tDNA on the TI plasmid. Okay, I hope this is very much clear that we do co-transformation of these two plasmids together in the Agrobacterium. This is a binary vector system. Now, does every Agrobacterium which we use in the laboratory needs a helper plasmid? Answer is yes. Some of the available bacterial strains, they already come with a helper plasmid inside them. So you don't need to do co-transformation. But some of the bacterial strains, they come without any helper plasmid. Okay, in that case, you have to co-transform. We are using AGL1 strain of Agrobacterium at FCC. Okay, so it requires BAL154, which is a helper plasmid. Okay, and then we have a TI plasmid, a PSP219 or PSP218, which requires that helper plasmid BAL154, which is carrying the virulence genes. And we have AGL1 strain, we have LBA404 strain. LBA404 does not have a Kamuri fragment in the genome, but AGL1 has got a Kamuri fragment on its genome. So what happens when we transform the PAL154, the TI plasmid, that is PSP219, into the AGL1, it becomes super virulent strain having the TI plasmid. So the efficiency of transformation is very much enhanced. You don't need to remember AGL1, you really don't need to remember LBA404, okay, or the name of any plasmid. You must know basic things like a helper plasmid, what it is carrying, and along with a helper plasmid, you need a TI vector, which contains the cassettes of your interest within the left and right borders, and you co-transform them into Agrobacterium. Okay, and then you use that transform Agrobacterium to infect bacterial cells. After infection, what you can do is you can take the plant cells to the growth medium, which contains the antibiotic in it. Only the plant cells which have acquired tDNA carrying kanamycin, for example, here, they will grow, all the rest of the cells will die. So this is the transgenic plant selection. Now remember another thing, because we are doing co-transformation, and I said this TI plasmid has got one antibiotic here outside the left and right border, which we use for the selection of transformed Agrobacterium. Do the helper plasmid have an antibiotic resistance gene? The answer is yes. It has another antibiotic resistance gene. So you can think here, there are 3 antibiotic resistance genes in this binary vector system. One here, and the third one here. Don't use this. We use this after transformation of plant cells. Okay, it is under a eukaryotic promoter. Remember that, and that promoter will only express in the eukaryotic system, the plant cell, not in the bacterial cell. So at least 2 antibiotics will be there in the medium for selection of the Agrobacterium, which has got TI plasmid plus the helper plasmid. One antibiotic will be for this selection, the other will be for this selection. Only the cells of bacteria which acquire both of these plasmids, they will grow on that antibiotic medium, that is double antibiotic. Sometimes the Agrobacterium may itself have antibiotic resistance gene in the genome of that. So, in that case, we need to go for 3 antibiotics. Okay, so don't be surprised if you hear that the plant transformation people, they use 3 different types of antibiotics for selection of Agrobacterium, which is transformed with a binary vector system. One of the antibiotic resistance might be on the bacterial genome, the other might be on the TI plasmid, and the third one might be on the helper plasmid. So you have to use all the 3 antibiotics so that the right bacterium with the right helper plasmid and right tDNA plasmid, only those should grow. That is one of the very important steps before you proceed for transformation. Okay, remember this.
Sir, can you please repeat the purpose of the third?
So antibiotic number 1 on the TI plasmid. Antibiotic number 2 on the herpal plasmid. Antibiotic number 3 in the genome of Agrobacterium. Okay? So when you transform these two plasmids into Agrobacterium, which already has got an antibiotic resistant gene, so you need to select the transformation mixture on 3 different antibiotics together, so that only the Agrobacterium, which is capable of growing on the 3 antibiotics, will be grown, will make the colonies. All the other Agrobacterium cells, they will die. Okay? And the one which will grow will have an antibiotic gene on the whole bacterial genome, which is obviously there. And then they have 2 antibiotic resistance capabilities, one from helper plasmid, one from TI plasmid. Is it clear?
Yes, sir.
So
Okay.
the helper plasmid lacks the multiple cloning site, right?
Yes, yes, because there is no need to clone in it because it does not have any left and right borders. Left and right borders are essential in making tDNA. We don't want the helper plasmid to produce a tDNA. We only want the TI plasmid to produce the tDNA because our genes of interest, they are here. Okay. If we generate a tDNA here, if we generate a tDNA here, then what will happen? That tDNA from the helper plasmid can get integrated into the plant cell. Okay. So our mission is to incorporate these genes, not any other gene on the helper plasmid or the TI plasmid. Only this part of the genes we want to transfer. And that's the reason we allow only this part to get single-stranded tDNA. Okay?
Okay, yes, sir.
Okay, so the transformation efficiency of the TI plasmid through an agrobacterium, it depends on, I have been mentioning, a host range. Even within the same plant species, you know that there are different cultivars of wheat. They're different. So many. Galaxy, Faisalabad 2008, okay? NARC 2011, Pakistan 13. These are all different cultivars of varieties of wheat, okay? So if you try to infect all these different cultivars, they're all actually wheat plants, but they are different cultivars. They will show you different transformation efficiency, okay? And so always keep in mind that the transformation efficiency is dependent on the cultivar and the tissue type. If you transform an embryo or if you transform a hypocotyl, the transformation efficiency will be different. Okay, so it means that the formation of tDNA and movement of tDNA to the nucleus is dependent on the cultivars and the type of tissues. So what you do, you optimize a method which best performs in your laboratory. Here at FC, what we do is that we isolate the embryos from developing seeds at 15 to 20 days post enthesis. Again, you know, we need to observe the plant every day, every day. As soon as we see the yellow color pollens on the spike, we say this is 1 DPA, and we label the plant that this is 1 DPA on this date. And then after 14 days, we take out all the seeds which are actually immature seeds. We cut those seeds and take out the embryos from there. Okay? We transfer 5,000 embryos for getting only a few transgenic calli. Can you imagine the amount of work that it requires? 5,000 embryos, and if you have to isolate 5,000 embryos, definitely you will get a spine pain. That's a big job, I'm telling you. The people here, who are using plant transformation, they sit all the day on the laminar floor in isolating the embryos, and in the evening, they just do infection. Then next morning, they transfer the embryos to antibiotic selection medium, that which as the embryos have been transformed. And from 5,000 transformations, what you get? 1, 2, 3 transgenics. And there's a long process of selection on antibiotic. Anyway, so I was telling you that the transformation efficiency is dependent on the cultivar and the type of tissue. Okay, some of the plants, they perform better on hypocotyl tissues, but wheat plants are usually transferred using the developing embryo. Okay, and isolation of this embryo from the 15 to 20 DPA old developing seed. Okay, so methods for plant transformation, other than the indirect method, they include one of the very important method, which is very well mentioned in the literature, and it is used for a lot of transformations. That is particle bombardment. The technique has 3 different names. One is particle bombardment, other is called Biolistic method of transformation. The third is called Microprojectile. But all these 3 mean the same. We are using the same gene gun, no difference in the methodology. But some people, they say particle bombardment because it uses the particles for the bombardment. Biolistic, I mean, there is a blast of gas here, so people say biolistic. Microprojectile, it's a targeting of tissues with micro carriers, so people call it microprojectile. But all these 3 terms may mean the same thing. What you see on the screen is actually a particle bombardment gun. This is not really a gun, but this is a machine, very simple machine, but very costly one. Okay, it's costly due to several reasons. The only manufacturer on the globe for this machine who has got the property rights for that is BioRad company. They charge about 35 million rupees for this machine. 35 million, you can imagine. This is a very simple machine, having 2 gauges, a chamber in it, 2 knobs, one for regulating the pressure, the other one, just you hit the knob to give the gas blast, okay? And you can see some pipes here, which are coming out of this machine, and I will tell you that these are all the other accessories which are required for transformation purposes. I will explain to you to familiarize you with this gene gun, because it is not something really big, it's very simple, I'm telling you, very simple. So there is nothing to be afraid. You can just go ahead and start using this machine. We are trying a lot to get this machine at FCC, okay? So, and hope sooner or later we will get it sometime. What it has got, this is the chamber that I showed you here, okay? And this chamber has got some pipes on it. These pipes, they are connected to a helium cylinder, okay? The helium cylinder has got pressurized helium gas, just like an oxygen cylinder or a nitrogen cylinder, okay? They also have gauges on it, and the pipe is connected toward on the back of this machine, okay, to supply the air pressure. It's not air pressure, it is helium pressure. In between these pipes, here and between the machine, there is a solenoid valve. This solenoid valve actually permits the gas to flow when you achieve a very specific pressure, okay, which is controlled by this knob, okay. You adjust this knob to reach 700 psi, 900 psi, or 1100 psi. There are three options for you, okay, because there are three kinds of rupture discs which are available. So what happens is, the process is very, very simple. You have a rupture disc. Rupture disc goes into this assembly. You can unscrew this assembly and you can find a space to place a plastic disc in it. That is a golden color, yellowish golden color plastic disc. Three types of discs are available. One can tolerate up till 700 psi. The other one can tolerate 900 psi. The third one can tolerate 1100 psi. If you are using very soft tissues like tomato or tobacco, they are very soft tissues. You use 700 psi disc because you don't need a lot of pressure to push the tungsten or gold particles into the cells. 700 psi is more than enough. If you are transforming cotton tissues, cotton is a hard plant. It requires a lot of pressure, force of the particles to penetrate into the cells. You go to 1100 psi. If you need wheat for transformation through Agrobacterium method, you need 900 psi is enough for that. It's not soft tissue, it's not very hard tissue. Okay, so now you understand that why we use different types of rupture discs. Rupture discs, they are in between this assembly and the gas pressure. As soon as the gas pressure reaches the set pressure for the disc, you have to set 700 psi using this knob, and when this knob goes to 700 psi, this rupture disc will blast because it cannot tolerate the pressure more than 700 psi. Okay? And then this is the main assembly which contains your macrocarriers and microcarriers. What is a macrocarrier? Can you imagine what is a rupture disc? It's a circular disc which is transparent but golden in color, and it can resist certain pressure. If it is meant for 700 psi, it's a very precisely built disc, okay? So it will rupture at 700 psi. The 1100 one will rupture at 1100 psi, okay? So you keep the rupture disc in this assembly, and this is a separate assembly just below that, not connected to it, but there is a big hole in it. If you unscrew this part, there is an option to put another disc, which is called macrocarrier. Macrocarrier is simply a disc on which you put the gold particles or tungsten particles coated with your TI plasmid or DNA having the cassets, or the D2, with D2 protein and the gene of your interest. You can coat the DNA with any of the DNA particles or protein particles. You just need different types of treatments. It's a simple procedure. You need spermine for DNA and high concentration of magnesium chloride. It will coat your DNA on the tungsten or gold particles. That's a simple method. Okay, and then you take some volume of that and put it on the macrocarrier. And that suspension of tungsten particle or gold particle, which are coated by DNA, they are suspended in absolute ethanol. And they are not allowed to stick with each other. The coating requires a practice where you ensure that these metal particles, they don't adhere to each other to make a single mass. They remain separated so that each separate micrometer in size, these tungsten particles and gold particles are. And we want single particles to enter into single nucleus. If you bombard and multiple number of tungsten particles, they enter into a cell, they can damage the cell and the cell can die. Okay? So you need to maintain a small concentration of the tungsten particles coated with the DNA, all of them separated together. You just drop with the help of a micropipette about 5 microliter of that mixture in the center of that macro carrier disk. And it immediately starts spreading and drying. So those gold particles or tungsten particles coated with DNA, they dry on the surface of macro carrier. Everybody, I think, in our childhood, we have played with sand. Okay? Sometime we want to clean our hands, water is not available. So what you do? You smash your hand over the other hand so that the dust or the soil adhering to your hand, it separates from your skin. Okay? The exact principle is used here. When this bursts, the disk here bursts, the helium gas strikes this macro carrier, and on the macro carrier, these tungsten and gold particles are adhering, just like the sand on our hands. Okay? Below this macro carrier is an iron screen, the same iron screen that we use in our doors. There's a small circle cut out of it, and the macro carrier strikes with that iron screen, releasing the gold or tungsten particles adhering to the macro carrier. All they start bombarding on the tissues which are there in this patty plate. This distance is movable. It depends what kind of tissues and what kind of pressure. It can be maintained by the distance of the rupture disk, which is here, and your sample, which is here. You just have to press the button and adjust this pressure. Put the rupture disk here, put the macro carrier here. Below the macro carrier, you put an iron screen, okay? And then you press the button and you hear a little sound, not a big one, just and then it is done. You take out this disk, this patty plate, having the K-line, and you can visibly see the gold particles concentrated at some points and then very finely hitting some of the tissues. Okay, most of the particles, they can get into nucleus. Some of the particles, they just go to the cytoplasm. Some of the particles, if they are adhering to other particles, they can also rupture the cells, damage the cells. But a single bombardment experiment results into a lot of different cells receiving the DNA-coated tungsten particles. And there is a chance that that tungsten particle has gone to nucleus. Once the DNA is there in the nucleus, it is dissolved from the surface of the tungsten particles, and by chance, it can get into the nuclear DNA, the chromosomal DNA, by homologous recombination or some other recombinational processes. That is just by chance. Okay. Now, I will show you that this is the rupture disk. Okay. And then this is the macro carrier. Okay. And this is the iron screen. and this macrocarrier is loaded with the tungsten particles or gold particles coated with DNA. And when you do the bursting of this rupture disk, helium gas pressure takes the tungsten particles, which are known as microcarriers. What is a microcarrier? It's a tungsten or gold particle carrying the DNA. It's called microcarrier because these are microparticles. They're just microns in size, even smaller than micron in size. Okay, they're called macrocarrier and hit the tissues. When they hit the tissues with the force, they can enter into the tissues. So this is the other diagram showing the same, and this is actually a picture showing that how microcarriers, the microcarriers, they are gold particles or tungsten particles. They flow out of the macrocarrier, pass through the iron screen, and they hit the tissues in the packing plate with a great force. Okay, so this is simply a particle bombardment gun. It's not very scientific like Agrobacterium transformation. It's just a simple equipment which you use for loading your DNA on the microcarriers, which are gold or tungsten particles, and enforcing those particles under high pressure, just like a bullet goes through high pressure, just like these microcarriers, they go with a high pressure of helium and strike the tissues in front of them. Equipment used in the laboratory, it is not a mobile. You just put this equipment in a laminar flow, okay? The cylinder is outside the outside the laminar flow, and you perform your experiment. There is another version of the same which is called handheld. You can take it to the field, to anywhere else, and you can do your experiment. So I'm ending this lecture today at this point because I have reached the time limit.